Picture walking into an emergency room. Maybe you've had a bad accident or, you're prepping for a major surgery.
Right, something where they need to know your vitals immediately.
Exactly. And the intake staff asks for your blood type.
Hmm.
When that happens, you probably conjure up a simple letter, right? Like an A, a B, or an O, and maybe a plus or minus sign.
Yeah, we treat it like this tidy little category, almost like a biological zodiac sign.
I mean, that's exactly how it feels. But what if that familiar label is actually just well, a vastly oversimplified shorthand, like a highly sanitized summary for this massive, unique, and densely layered chemical fingerprint that's basically coating every single one of your red blood cells?
It genuinely is a dense fingerprint. We are so conditioned to view blood typing as this binary checklist, but the reality playing out on a cellular level involves a highly intricate forest of molecules.
Which is really the core mission for our deep dive into the sources today. We're going to decode the ABO and Rh systems from the ground up.
It's a fascinating history, too.
Oh, absolutely. We're going to uncover what actually determines your blood type, right? And we'll explore why mismatched blood acts like a literal poison in the body. Plus, we'll look at some of the fascinating genetic exceptions that completely break all the medical rules.
Just to set the proper context, though, before we jump into the mechanics, the International Society of Blood Transfusion actually recognizes under 40 distinct blood group systems.
Wait, 40?
Yeah, over 40. There are systems with names like Kell, Duffy, Kidd, Lewis. But today, we are focusing on ABO and Rh because those two are the absolute heavy hitters. I mean, they're the ones that dictate survival in clinical medicine.
Right. And to understand those heavy hitters, we really have to look at how we discovered them in the first place, because it wasn't always a neat science.
Oh, not at all.
We're talking about Vienna, around 1900 to 1901. Before this, giving a patient a blood transfusion was a total toss-up. It was essentially playing Russian roulette with someone's life.
It really was. One patient might recover beautifully, and the next would they'd experience violent fever, complete kidney failure, and death. And no one had the faintest idea why.
That is just terrifying. So how did we finally figure it out?
Well, it comes down to Karl Landsteiner. He was an Austrian physician, and his experiment was embarrassingly simple. Brilliant, but simple.
How simple are we talking?
I mean, he didn't use complex equipment. He basically drew blood from himself and 5 of his colleagues. He separated the liquid serum from the solid red blood cells, and then he just systematically cross-mixed them on a matrix.
Just mixing everybody's blood with everyone else's.
Exactly. Every person's liquid serum introduced to every other person's red cells just to see which combinations clumped together or agglutinated and which ones stayed smooth.
Okay, let's unpack this. It's like trying to jumpstart a car, right? But sometimes the jumper cables arbitrarily decide to melt the engine. Landsteiner essentially created the very first compatibility chart.
That's a great way to put it. He mapped out 3 distinct biological profiles based on what melted the engine and what didn't. He called them groups A, B, and C.
Though the sources note that group C was quickly renamed O, right? From the German word ohne, meaning without.
Right. And then his colleagues, Di Castello and Sturley, found the AB group about a year later.
Which you'd think would be this huge parade-worthy moment in medicine. But one of the most surprising facts from the sources is that the medical world basically ignored this for 3 decades. Landsteiner didn't win his Nobel Prize until 1930.
Yeah, the delay is wild, but it makes a dark sort of historical sense. Surgical techniques back then just didn't allow for mass transfusions anyway.
Oh, I see. So there was no practical application yet.
Exactly. It wasn't until World War I, where battlefield surgeons were dealing with massive hemorrhagic shock in soldiers, that ABO typing became an absolute desperate necessity.
Wow. Okay, so now that we know how it was discovered, we need to understand what Landsteiner was actually looking at.
Yeah.
What is physically clumping in those test tubes? Like, what is the anatomy of a blood type?
It all comes down to antigens. You can think of an antigen as a little structural marker sitting on the outer membrane of your red blood cells.
Okay, like a name tag.
Sort of, yeah. Your immune system is like a bouncer. It constantly scans these markers to figure out if a cell is self, meaning it belongs there, or foreign, meaning it needs to be destroyed. And in the ABO system, these markers are actually sugar chains.
Wait, sugars. The thing dictating whether a transfusion is safe or lethal is just sugar.
Basically, yes. And it's all dictated by a gene on chromosome 9. Now, everybody starts with this base scaffolding on their cells called the H antigen.
Oh, okay. Let me try an analogy here. Imagine the red blood cell is a military uniform, and the H antigen is just a blank square of Velcro sewn onto the shoulder. So everyone has the Velcro.
Yes, perfect. Now, if you inherit the gene for type A blood, your body takes a very specific unit badge, a sugar, called N-acetylgalactosamine, and sticks it onto that Velcro.
Right.
If you're type B, your body uses a different sugar, galactose, and sticks that badge on instead.
And if I'm type AB, I get both badges on my Velcro.
Exactly.
Huh.
Which leaves type O. If you're type O, your gene is functionally inactive. You add nothing. You just have the bare Velcro, the unmodified H antigen.
Okay, but here's where it gets really interesting and frankly a bit puzzling in the sources. If I am type A and I've never been exposed to type B blood in my entire life, why does my body instantly have the antibodies to attack it in a transfusion? How can my immune system know to fight something it's never met?
That is the big counterintuitive marvel of clinical medicine. It's governed by Landsteiner's law, which says you automatically carry antibodies against whatever you lack.
But how?
Gut bacteria.
Wait, really? Bacteria in my stomach?
Well, your intestines, yeah. The common bacteria in your gut naturally carry antigens that are structurally identical to the A and B sugars. So from the time you're an infant, your immune system is exposed to them. They basically vaccinate you.
Oh, wow. So if I'm type A, my immune system ignores the A-like bacteria, but sees the B-like bacteria and goes, hey, that's foreign, and builds antibodies.
Precisely. You are pre-armed. That's why a mismatch is so catastrophic. Introducing just 10 to 15 milliliters of incompatible blood, which is like barely a tablespoon, causes an immediate fatal hemolytic reaction.
Because the weapons are already circulating.
Exactly. The donor cells instantly clump together, jam up your kidneys, and literally rupture inside your blood vessels.
That is brutal. Okay, so that's the letters, the ABO part. But what about the positive or negative sign, the Rh factor? The sources make it seem like the Rh rules operate on a completely different biological logic.
They absolutely do, and the discovery of Rh was incredibly messy. Between 1937 and 1940, Landsteiner and Alexander Wiener found this factor in rhesus monkeys.
Right, but there was also this tragic human case happening at almost the exact same time, wasn't there?
Yeah, in 1939, two doctors, Levine and Stetson, had a patient who had a stillborn baby. She was hemorrhaging, so they gave her a transfusion of her husband's blood. Now, the husband was a perfect ABO match.
So it should have been completely safe?
According to what they knew, yes. But she had a massive, severe reaction to it. They realized her immune system had reacted to an unknown factor in the fetus's blood, something inherited from the father.
Wait, if ABO antibodies are automatic from infancy because of the gut bacteria, why did this mother only react after having the baby?
Because the Rh factor is a protein, not a sugar. Your gut bacteria don't have Rh proteins, so if you are Rh negative, you do not spontaneously carry anti-Rh antibodies. You only develop them if you are physically exposed to Rh positive blood.
Got it. So the immune system actually has to touch the Rh protein to know it's bad.
Exactly, and that usually happens either through a bad transfusion or during childbirth when some fetal blood crosses into the mother's circulation.
But by the time she builds the antibodies, the first baby's already born, so it's fine.
Right. The danger is the second pregnancy. If she has a second Rh-positive baby, those antibodies will cross the placenta and attack the fetus. It's a condition called hemolytic disease of the newborn.
Which brings us to one of medicine's greatest triumphs, right? Rogam.
Oh, absolutely. Developed in the 1960s, it's a shot given to the mother that basically mops up any stray fetal Rh-positive cells before her immune system can even see them. It prevents her from ever getting sensitized. It has literally saved hundreds of thousands of infants.
That's essentially like an invisibility cloak for the fetal cells. It's brilliant. But I want to touch on this bizarre naming dispute in the sources. The monkey factor and the human factor weren't even the same thing.
No, they weren't. They just looked similar in primitive tests. They eventually had to rename the monkey one the LW factor, but the human one kept the Rh name.
And because of that chaos, we're stuck with this dual naming system today. The sources mentioned the Fisher race system, which uses C, D, and E, and the Wiener system, which uses terms like Rh1 and Rh2.
Yeah, it's a classic rival framework in hematology. It's mostly Fisher race now, but you still see both in textbooks, just scientific baggage from when discovery outpaced organization.
Classic. Okay, so biology really follows tidy rules. If ABO and Rh are the strict laws of blood, we really have to talk about the rule breakers, the people whose bodies just completely defy these laws.
Oh, the exceptions are fascinating.
Let's start with the Bombay phenotype, discovered in 1952. Patients tested as type O, but their serum weirdly attacked true type O blood.
Which shouldn't happen.
Right, so what does this all mean? If the H antigen is the foundation of a house and the A and B sugars are the walls, are you saying these folks lack the FUT1 gene so they can't even pour the foundation?
That is exactly it. They have no H antigen at all. Their red cells are completely bare.
So they literally don't even have the Velcro.
No Velcro, which means they have anti-H antibodies. If you give them regular O-negative blood, which has the H antigen, their body sees it as a foreign invader and attacks it.
Wow, so that totally breaks the rule that O-negative is the universal donor.
It absolutely does.
And then there's the extreme opposite end of the spectrum on the Rh side, the Rh null condition, or golden blood.
Yeah, golden blood. These people are missing the entire Rh antigen complex, not just the positive and negative part, but all the related Rh proteins. There are fewer than 50 confirmed cases worldwide.
Wait, 50, like 50 in the whole world?
Yeah. It is incredibly rare. They are the true universal donor for Rh systems, but it's incredibly dangerous for them to have it. They can only ever receive blood from other Rh null donors. Plus, missing those proteins makes their red cells structurally fragile.
That sounds like a terrifying biological superpower. So because blood types are this complex and so deeply ingrained in our biology, it seems like humans just naturally try to find meaning in them.
Right. Oh, people love to find patterns. Sometimes it leads to pseudoscience, but other times it actually reveals real evolutionary connections.
Let's do the pseudoscience first. The sources talk about the blood type diet from the 90s, and the Japanese Kitsuikigata, the idea that your blood type matches your personality.
Yeah, both of those have been thoroughly debunked. There was a massive PLOS-1 study in 2014 that completely dismantled the diet claims. Eating vegetables helps everyone, regardless of blood type.
Shocker.
Right. And blood type is purely a biological classification of red cell surface antigens. There is zero established link to your psychological traits or your nutritional needs.
Okay, fair enough. But let me push back a bit. If it doesn't dictate my diet or personality, does my blood type actually matter when I'm not getting a transfusion?
Yes, absolutely. Because these antigens actually overlap with other bodily systems.
Like what?
Well, clotting, for example. Type O individuals naturally have about 25% lower levels of something called von Willebrand factor.
Okay, and what does that do?
It helps your blood clot. So having less of it means type O people have a lower risk of dangerous blood clots, like venous thromboembolism, but they have a higher bleeding tendency if they get injured.
Oh, wow. Okay, that's a very real physical effect.
And it goes deeper. Let's look at malaria. Remember the Duffy blood group?
One of the 40 other systems, right?
Right. Well, the Duffy antigen acts as a literal receptor for a specific type of malaria parasite, Plasmodium vivax. It uses that antigen like a doorway to invade the red blood cell.
That is wild. The parasite evolved to use our own blood markers against us.
Exactly. But humans evolved right back. Today, many people of West African descent naturally lack this Duffy antigen entirely. The doorway is just gone, which gives them a natural resistance to that strain of malaria. It's a prime example of direct evolutionary selection.
That's incredible. And didn't we see something similar with COVID-19?
We did, yeah. Observational studies suggested that type A individuals had a modestly higher risk of severe infection, while type O seemed somewhat protective. The exact mechanism is still being figured out, but it shows how these antigens interact with modern diseases.
So to synthesize all this, I mean, what a journey. We started with Carl Landsteiner in a lab, just watching test tubes clump together, finally ending the era of blind randomized transfusions.
A huge leap forward.
And then realizing that type AB and type O aren't just points on a spectrum. They're these perfect combinatorial mirror images, right? AB has both antigens but zero antibodies. And type O has zero antigens but carries both antibodies.
It's beautifully symmetrical. And, since you mentioned type O, it's a good time to remind everyone about that universal donor misconception.
Oh, right, the O-negative rule.
Yeah.
Oh.
People think O-negative whole blood is perfectly safe for anyone, but that rule only applies to packed red blood cells.
Because the whole blood still has the plasma, and the plasma has the antibodies.
Exactly. If you give someone O-negative whole blood, you're pumping them full of anti-A and anti-B antibodies. It's highly dangerous. You have to separate the red cells first.
It just shows how precise all of this has to be.
Yeah.
And, it leaves me with this thought. You walk into that ER, they ask for your type, and it's just this tiny little shorthand.
Yeah.
But your blood type is genetically fixed for life? barring a rare bone marrow transplant.
Right, it's yours forever.
And we know that antigens like Duffy evolved specifically to fight off malaria over thousands of years. So it really makes you wonder, what other invisible evolutionary battles might your specific blood type be quietly fighting or even remembering inside your veins right now?